Hey guys, welcome back. We are dealing with the the last two main methods. I actually added another one. I'm not sure if I told you how many, but I added uh, two more to look at. We'll be finishing up now our protein interaction techniques with this lecture, and uh, probably starting on the test 3 material next time. So I think this block will be the last material. We'll talk about that Tuesday, but and our test will be coming up shortly. I think I'll have to figure it out exactly when things are happening here. But anyway, I think this is a good stopping point. I don't know. I hadn't decided yet. But for right now, this is the last of the protein interaction technologies anyway. Uh, this is called a far western. And so we had certainly discussed western blotting. And if you remember, we went through the SDS page, the 1D, the 2D, the denaturing of the proteins, the electrophoresis, the blotting technology where we moved the proteins that we had separated out in 1 or 2D and actually moved them to a piece of nitrocellulose or nylon that then we were able to probe in a Western blot with antibody, and then usually a secondary antibody to the primary antibody was used with some type of detection molecule attached, whether that's an enzyme, a radioactive molecule. It depends on sensitivity and cost, whatnot. So a Western blot is an antibody to a protein. A far Western, though, is a modification of the Western blot, and in this case, it's used to detect protein-protein interactions. And Western blot again uses the antibody, and this is to detect, the protein of interest. The far Western will use a non-antibody protein, which can bind the protein of interest. So this could be the bait protein, the bait, and we could be testing various amounts of prey proteins in this technique here. So Western blot is an antibody used to detect certain proteins. Far Western will be looking at the protein-protein interactions, the bait to the prey. So again, in protein-protein interactions, there is this multi-technique required. So this is just another one of these techniques that one might use to further piece together or confirm two proteins do interact here. So how does this work? Here's a cartoon, and basically they take a GST tagged protein X, and we'll call that our bait protein, our bait. Okay, so bait protein here. Okay, so protein X is our bait. And we do in vitro kinase reaction. And in this case, that labels the bait protein with a radioactive phosphorus. And this is the little star here. And the labeled protein is then you remove the GST tag, and now you're left with the protein that's radioactively labeled. And for right now, we will just say that in molecular biology, we will discuss screening here a little bit. Molecular biology. Okay, looks like a dog, doesn't it? Okay, molecular biology. So we're not going to hit that, but here we could then separate, say, cells we don't know if there's a prey, but here are cells that are non-dividing, quiescent, G0, and here are cells that are actively binding. And we then probe the Western blot. Here's our protein markers. With this radioactive protein, you allow it to bind. And this, of course, probably is native SDS, so it won't be denaturing. And in this case, in cells that are not dividing, we see one protein. And that same protein is in dividing cells, but we see a second protein or a second prey only in cells actively dividing. Okay, so this could be wild type cancer cells, etc. And we're looking for proteins that are the prey for the bait. Here they are, protein X, and proteins that might be different. And of course, if we had a known series of preys, we could put those into other lanes and ask the question, does the protein X detect those as well? So that's a far western. This is just for your knowledge to compare western and far western here. And this is some company had this selling a kit. Here's the protein separated by electrophoresis and transferred to the membrane or blotted. And then the bait protein is labeled with some, this could be radioactive, could be an enzyme, and then you allow the probe to bind, okay? And then if it's non-radioactive, then you might even have an antibody to the ligand, and then that is maybe an enzyme, or it looks like they've got a horseradish peroxidase, and they add the substrate for that enzyme, and you get light produced. So if you expose to X-ray film, you would find this protein was detected by this labeled bait protein. So we found our prey here. And if this ligand is radioactive, then it will be exposed on X-ray film. And again, there it is. And you could purify this protein, probably not in a 1D. I would definitely want to do a 2D gel. Okay, a 2D gel to try to purify this. And usually far western is not the first choice, but it could be. It could be. And then we would do 2-hybrid co-immunoprecipitation pull downs to test that further. So that's the far western here. And so this is just discussing how we will do a general overlay assay. And so in some ways, a far western is an overlay assay. And so it's called a far western. It's also known as an overlay assay. And again, you're not using an antibody, even though they're saying that you are. It's usually a protein. And here they're just using biotin to put on the bait and detect a strep conjugate. But biotin and strep are so efficient that that can have a problem in prey interactions here. So a lot of times they'll actually radio labeled here. And so each prey is immobilized and then tested with your far western bait protein as your probe here. And so what does that look like? Well, here's a far western overlay of a GST tag adrenergic receptor, carboxy terminus. And what is going on is I don't know if you've had physiology, but there are multiple flavors of adrenergic receptors that are involved in human physiology. And in this case, it's AR1 C-terminus AR2. So these are two adrenergic receptors, but they are not the same protein. They're very related, but not the same. And so in the lanes, all these letters and numbers are different proteins that they're testing. And a lot of these proteins have a known domain that binds receptors, PDZ domain. And all these different proteins and parts of different proteins have this domain in it. And so equal amounts of tagged proteins. So each one of these was tagged with HA, the Ha fusion. They purified them and then added it to page gel, electrophoresed it, blotted it, and then it's called an overlay. Then you overlay with your far western labeled protein here, and in this case, this one up here in the red circle is number 1 AR, and that's at the end of the protein. So they must have some evidence that the C-terminus of this adrenergic receptor 1 binds potentially to PDZ. And so they compare all, let's see, 1, 2, 3, 4, is it 5 different proteins or 6? Anyway, they compare 1, 2, 3, 4, 5, 6, 7 actual proteins. And right away you see that the bait has bound to 3 of them. So overlays with the B1 adrenergic receptor, and that's right here, it's the B1, reveal strong binding to this protein, MAGI2 PDZ1, MAGI1 PDZ1, so it's MAGI2 protein, MAGI1 protein, and PSD95 PDZ73 protein. Boy, that's a big names. And so within the red circle, we see now positive detection by our overlay here. So right now we can say these 3 proteins must contain domains that bind to this receptor under the conditions we're testing. The other proteins are found not to bind here. So you see there's no bands here. So these are non-binding. And so when they look at the next protein, B2 adrenergic receptor, long story short, it recognizes this protein only. So it's quite different from its cousin, B1. So these are 2 related adrenergic receptors, but they have different targets here. And again, this is shown a far western overlay assay. And so here's just taking this a little further. This is where we do reciprocal experiments. Okay, key concept here. If your overlay finds the bait, finds the prey, then you should be able to flip and label the prey and find the bait. The word reciprocal means an exact flip. So in this cartoon, they put GST by itself, GST B1 adrenergic receptor, B2 adrenergic receptor, and I'm not sure if there's another type of adrenergic receptor here. I'm not sure what they're showing. And anyway, they used the MAGI2 protein, which was the found prey now, as a probe on your overlay, and it confirms that it binds to the B1 adrenergic receptor only, not B2. So the interaction between B1 and MAGI2, this domain, can be visualized in either direction or either reciprocal experiment. If you didn't see this, that would be very concerning, that the previous something was wrong, and you would have to redo it until you were, sure about what you had found. So you can see now that the overlay can find new proteins, or it can confirm proteins that are, say, predicted by bioinformatics to interact together. And we see what's going on here. So again, reciprocal experiments. So in some ways, this is a control. Your reciprocal must confirm what your other overlay showed. And here's just GST by itself, so there's no interaction here. Okay, so reciprocal experiments to overlay assay. And this is probably more biochemistry, but once they knew that these two proteins interact, they actually tested in nanomolar amounts of this magi how much could a certain amount of adrenergic B1 adrenergic receptor bind to it. So they had strips containing 2 micrograms, and they were incubated with the magi. Okay, so they overlaid it, and they did it at 6 concentrations between 1 and 300 nanometers of the detector here, the Magi 1. So they did a low level, so there's almost no detection here. And the highest level was up in this region, and you see we'd already hit a plateau. And so binding did not decrease. Basically, it peaked at 100 nanomolars, and the 300, there was no real big change here. And so they consider 300 nanomolars as the maximum amount of magi that can bind to 2 micrograms of pure adrenergic receptor. So you can see right away that you can actually test the binding of affinity and the amounts of 2 proteins coming together as well. So our previous work showed that the proteins can interact. This goes a little further and says, well, how much is that interaction? And it's probably getting close to what actually is occurring in the cell. So you could imagine a disease situation where one or the other partners had a low affinity, maybe could bind a little bit. And I mean, if you didn't bind at all, maybe it was lethal. So we wouldn't be born. But maybe if we had a lower affinity, we had a disease or were just sick, but still could kind of make it through a little bit. So I think in a personal genomics, what they're going to do is not only figure out your enzymes, but they're going to make predictions as to how strong or weak your enzyme interactions and your receptor interactions are. So you'll be further characterized throughout all your receptors and interactions there in deeper amounts, deeper details, and try to correlate that to diseases. Okay, the final protein interaction technology is called fluorescence. I'm sorry, FRET. It's for some name for the energy. I always thought it was fluorescence electron transfer, but there's some word that the F stands for. It's no big deal. Everybody says FRET. And so the concept of FRET is this is inside the cell. So this is in vivo. Okay. So now you see we have another technique that's in vivo. This is real time, kind of like the 2 hybrid system is real time. This is real time. And we take protein A and protein B and tag them with 2 fluorescent molecules that are different. One is releases in the cyan or blue wavelength, and the other one is in the green wavelength. And long story short, if the prey and bait are bound with their 2 fluorochromes, What happens is when you excite the blue with its wavelength, and that's what they're trying to show here, instead of getting a blue color released, okay, and you could see that under the microscope, you do not see a blue color. What happens is the energy of the electrons is donated over here and excites the green fluorescence. And if there's bait and prey bound, then if you look at them under a fluorescence microscope under FRET conditions, the inside of the cell where these proteins are will be green. If A and B do not bind and you excite it with blue, then you would only see where A was because it would be lighting up blue. Green would not be visible unless you excited at the green wavelength. And so you start with blue, but you end up with green. That means the bait and the prey are bound. You excite blue and you get no green. That means A and B are not bound. And they cannot be bound under that condition, under the disease, under the drug treatment, under the stress. It could change. It could be transiently changed. A and B could come together, then go apart. So if you had a movie, you could actually watch the fret and the energy transfer, and then it would go away again. And so here's a space filling model showing the excitation in the blue wavelength. And if they're separated, then you just get the blue color and there's no green fluorescence, okay, if the bait and prey don't interact. But if they do bind each other, and this is supposed to be the bait and prey up here, then you will detect green. Okay, it's the same thing as this, just showing the space-filled model. This is an in vivo test, in vivo. Okay, so when I ask you the big question, tell me everything about protein interaction technologies, that'd be a whopper question. Do realize there are in vitro and in vivo techniques. So here's another one here. This is, they've got membrane attached proteins, and it looks like this POI2, whatever that protein is, apparently there's some type of other protein. They bind to that, and then here is the yellow fluorescence. So you get a yellow color if you excite this at the yellow wavelength. And then there's this other molecule. Oh, that's number 2. This is number 1 with a turquoise color, so it's more a bluish, not yellow. So when we do 440 nanometers, we get a blue color if 1 and 2 are not bound. When you add this drug, rapamycin, they actually this causes a signal or a release of ions, and these two come together. That puts these two fluorochromes next to each other. You excite it for 40 nanometers, and now this area has a yellow fluorescence. Same wavelength, okay, different fluorescence. So you could see if you were watching a movie and you had these cells and you were zapping them with 440, the cell areas would probably be blue where the proteins were, little dots of blue. You added this drug to the cells, and all of a sudden those blue over time would disappear, and they would in fact become yellow. And so you can actually watch that over a time. So it's kind of a time course that you can watch the FRET here. And here's some, and so they have used this for biosensors. If protein 1 finds protein 2, that's sensing it. So it's telling you, hey, I found the bait and prey have found each other. So here's the sensory domain, and if it binds the substrate, here's the substrate, so they're not bound, so there's no FRET. When they are bound, then you're getting the green color. So they're saying blue is negative, green is positive here. So this is some type of enzyme with its substrate. When they find each other, FRET. Here is a century ligand with some type of domain, that a protein that binds a ligand, and that causes a shape change. And that shape change now gives a positive fret, and that can be uh, cobalt, could be zinc, could be hemoglobin iron, and you can watch the binding here, and you can monitor that in real time by a fret detection. Here's a domain with a substrate, and when that molecule is released, that's kind of held this square in check. The square is now free to bind into the domain, and you get a positive fret. Here is the opposite. So this is everything here. A positive gives fret. Here you start out with a fret, and if there's a protease that cleaves the protein here, then you release the protein and there's no fret. So you would lose the yellow signal in the presence of some protease. So you could detect a protein that's getting chewed up because you would lose the fret signal. So this is a fret, and you see that under certain, looks like they're activating SARC with hydrogen peroxide, and you're looking live imaging of the cell, and then I guess they add the hydrogen peroxide at T0, and at 20 minutes, it's hard to, well, it shouldn't be hard to see. It's bright green, and you can start seeing some red around the edges, and by 50 minutes it's completely red. So you've completely turned the fret from the green to the red channel over 50 minutes here, and they're just mapping that. Okay, so untreated, no change. Untreated would just look like that. Treated would give a positive fret signal there. Uh, this is the same thing. Looks like they've got when the two molecules they're trying to detect are not bound together, you're getting green. If they are bound together, you're getting a yellow color. And they show so here is the green and the blue. So I guess this is, I don't know what marker, what fluorescence they're doing there. But long story short, you look over a time course and you see right here, they add a growth factor. It's called epidermal growth factor here. Within 10 minutes, look at the fluorescence change in the cell. So within 10 minutes, this has occurred. And as you know anything about signaling in the cells, you don't want it to last forever. So what happens is after 60 and 130 minutes, the cells are back to the way they were. So you have a transient interaction, and then over time it goes back. And so it's kind of like the trap is reset again for some more interaction. And I thought this was neat, actually showing this going back to the originally where it started. But you can see after 10 minutes, this growth factor has caused the formation of this molecule. So we actually got a phosphorylation. So this is phosphorylation has occurred, and the phosphorylation, the phosphate goes right here, and the P means it's there. And then over time, so looks like somewhere between 10 to 60 minutes, some other enzyme comes in and takes the phosphorus off, and you go back to this state, and by 136 minutes, it's completely dephosphorylated until the next process to occur, and that's called fret. So you can actually monitor, so you can give drugs, treatments, you could test disease. Maybe your cancer cell doesn't behave like this. Maybe the cancer cell's never interacted. Or maybe the cancer cell starts out like this. It's always kind of on, and it never goes off. Okay, so fret is a way for you to test that. Okay, well, that's the end of my cartoon. I do want to say that if you had a protein going into an organelle, if you had a fluorochrome and there was a receptor that bound, say, the targeting, you might be able to do a fret to actually watch the proteins interacting real time with the receptor going into the organelle, in case you had a inquiry like that. Okay, so I think this is the end of the protein interaction technology lectures here. I hope you've enjoyed it. We will move on to other topics. Come back.